Okay, so another talk, which is mine this time. So that's pre-publication data sharing. And that's something I would like to share the results. They are quite still preliminary, but we are really excited to work on this. So if you think about pre-publication data sharing, right? So we have all agreed that data sharing is important, and we do that at the publication, right? Do we do it from the will or because of the pressure coming from the journal? I don't know, but we do that. But if you think about early data sharing, meaning you would do it early that you would publish the paper, it can have a benefit. It can enhance transparency. It can be independent scrutiny, meaning you will have access to the data before the paper is published, so you can verify the results and so on. And perhaps can promote responsible practices. There are concerns of the publication sharing, obviously fear of being scooped, maybe the data readiness is not filtered or was not prepared in the right format. Incentive is here, right? So like, what will be the incentive to do that? Because it requires substantial resources. And culture of not doing that and doing that at the publication time. So what we were curious to know is the current degree on which researchers share the omics data before the publication. So we took all the published preprints, and we thought if people share the preprints, and that's actually a great example where now it's widely adopted, and I think anybody who raised the voice against that will be not well received, meaning there is a consensus. And I think it's a great example how the consensus and the community have changed the norms. So it's a norm to share your research as the preprint, but it's yet the norm to share the data with your preprint. So what we did, we took all the preprints there, and we look at the published one, because we want to make sure it's published, and we look at the ones which did have GeoID inside. So we identify almost 7,000 preprints there. So what we do next, we see there is some interesting patterns happening. There are multiple GeoIDs there and so on. So what we did for now, as the preliminary results, we have simplified and said we would like to have the same GeoID in the preprint and the publication. And also that needs to be primary analysis. Remember the previous talk? So we don't want secondary analysis because there is no data generation there. So we ended up almost 600 preprints pairs there. So what we did next, we looked at the geo submission date. This is when you initiate the submission, but you can keep the data private, and public release date, when the data becomes public for the community. So we compare those two. So we said, what is the delay between you start your submission versus data becomes public? So there is around 8 months delay between you initiate it and keep the data private. So in other words, you keep your data, generally people keep the data private for 8 months. Then people do share, as they post the preprint, meaning the analysis is done, there is a 5 months delay before the data becomes public. And then what people do, they make the data public just before the publication. Perhaps when the paper is accepted and there is some process being initiated and request is there to share the data. So this is 1 month before the publication, people tend to make the data public. So what we see, we see some patterns of behavior or how the trends there. So the majority of things, the data is public before publication. So the way we define before means more than 1 week before the publication. But interestingly, the data was submitted before the preprint, but was kept private. So that's majority. Another category, public at the time of the publication, meaning 1 week before or 1 week after. That's 1/3 of the papers. And then the green category is public before publication, but submitted after the preprint. And there are usually multiple versions. So people submit first preprint, don't mention the ID, keep it blank, and then there is a follow-up version of the preprint where they put the GeoID. So one advantage to submit but not make it public is that you get your unique GeoID or SRID, which you can include in the preprint. And there is tiny fraction, for around 4%, which data becomes publicly available at the time of the preprint. So only 4%. So we have compared 4% for the rest, and we compare the citations, and our preliminary analysis suggests that if you share the data at the time of the preprint, you might receive increased number of citations, which can be a good incentive. Okay, so to wrap it up, so we see many researchers hesitate to share our omics data before publication, despite the preprint use. So I think there is a need to reshape the form of how to emphasize the benefits of early dissemination, which can accelerate the research. For the future work, we would like to compare all the publications there and the ones using the preprints. And we hope to see, or we will be curious to see if the people sharing the knowledge as the preprint, they tend to share the data early comparable to the rest. So acknowledgement, so many people being involved in both projects. And here it's collaboration between Georgia State University, Grigori Boldarev, there, Sankruti from UC, University of Suceava, Technical University of Moldova, and SAGE. An acknowledgement for the both project, the money we received from the EU, distributed to Romanian government. So that's the money for the research. And yeah, so we do cool science at SAGE, and we are eager to partner or talk about science. So happy to collaborate and partner. Okay, thank you.
Thank you for your very interesting talk. I really like the analysis of the publications. My question is technical. How do you assume that the paper is using a data set? How do you provide?
Yeah, so our assumption is that if they mention the data set ID in the data availability statement, that means they're using that, or they're generating. Yes, yeah, right. Yeah, sure.
Hi, I was wondering how you work out these papers getting citations when they already have geosource. Is it because they already have the data available that is actually why they're getting most of the citations? Or are they from lab groups that already have really high standards, in which case they're more likely to be cited?
Some groups, say again, please. Some groups are what?
They have already high standards of what they're doing, in which case they're just doing better science.
Yeah, no, that's a good question. So we should account for that, but probably will be hard to do it formally. Maybe consider H index of the leading or last offer, something like that. But we can try to, that's a great idea. Try to account that is not driving the signal. Yeah, thank you.
Thank you, good talk.
Hi, Serghei. I'm always fans of this topic. On your the first talk, the data sets that basically were not used, right? The kind of like unused data. Have you managed to drill down on to try and figure out, are there relationships with things like metadata richness?
Yep.
Are they less discoverable? And less contextual information?
Sure.
Things like that.
Yeah, no, thank you, Scott. So yeah, great question. So indeed, we didn't go much, but what we see, there are some interesting stuff, like for example, sample size. So nobody will reuse the data if your sample size is less than 10. That's clearly. And then absolute technology, like nobody cares about solid, if you remember solid, and I don't know how many people remember solid in this audience, but nobody cares about solid anymore. So things like that. So there are definitely features of the data which can predict. And I think this is the direction I'm really excited about. Try to predict the reusability from some features, like you mentioned, metadata availability and completeness and correctness and so on.
It is a sweet spot then, like so size, right? If it's too small, then it's not interesting, but if it's too big, then you can't even...
Oh, yeah, I guess so, right. And then maybe people take...
What is the optimum?
Yeah, right. Like, yeah, maybe you cannot reuse the entire TCGA, but you will take subsample of it and so on, right? But yeah, no, that's something we are really interested to dive in and see more insights. Yeah, thank you.
Hi.
Yeah.
I really love the stuff that you've done here. So I have some meta questions to the meta science. Will you release or share this data that you're collecting?
Yeah, no, absolutely. I think the plan is...
There's a great interest. There's lots of follow-up questions.
Yeah, no, absolutely. No, we would like...
With the each factor, obviously, you could say, okay, it's maybe the impact factor of the journal where it goes out, is it correlated? Or
Yeah, no, absolutely. There are many things which can correlate. But I think the ultimate goal for us, first of all, to publish it, I guess, but also to create the website or some sort of portal or leaderboard. And hopefully that can be dynamic. So people can come and say, what are the most data sets in this disease? Or what is the most data set for NIC? What is the valuable things people do? Right? So it could be like, and then I think cherishing, kind of hoping to introduce this like that index, reusability, what I call R index, whatever we call it, where hopefully can be somehow adopted, because it's not just number of citations you're getting, right? But hopefully, if you generate some data important for the community, you can use that index, and hopefully people can put it in CV or somehow use it for promotions.
Do you have any comments regarding the fairly large negative numbers that came out, or positive, depending on which way you look at it in your charts? You know, situations where you expect people to do something before or afterwards, there were quite some large outliers in the other direction. Do you have any comments on that?
Yeah, I mean, we didn't look carefully. I think what we saw, we did. I mean, probably I would not comment. We didn't explore that much, but those are interesting things. So, I mean, yeah. So for example, I think we saw somebody was, just like not publishing for a while, meaning they did the preprint, but was a delay in publishing for a couple of years or many years, and then during the time they did not share the data, for example. That's one example, but I don't recall others. But there is some interesting stuff there for sure.
Thank you.
Yep, thank you.
Last question from
Oh, yeah.
the other side.
Hey, thanks for the nice talk.
Sure.
In the first talk, you mentioned like thinking about data formats and uploading maybe processed data
Yep.
formats versus raw data. Isn't there value in uploading raw data in terms of long-term?
Yeah, no, absolutely. I don't doubt value of raw data, because this is the and if you think about the goal, what is the goal of SRA, of those public repositories? I think the primary goal is to save the data for generation, and the raw format serving that perfectly, no doubt about that. But I would like to challenge that and say that if you think about data reuse, raw data format is not the best, and maybe we can find the compromise. And the simple example, again, is the BEM file. So if you have BEM file containing both mapped and unmapped reads, you can get your FASTQ file from there. But at the same time, BEM file can give you easy access to reads, like, what is called slicing. You can slice reads and get the reads from HLA side, whatever your favorite gene or chromosome or unmapped reads and so on. So you don't need to run that redundant read mapping step. And you might say, the reference can be a concern, but we can think about how to update the BEM files and so on. So, but it's an interesting question, which I don't see much discussion at all, but I think needs to be there if we would like the data to be widely reused. I think that's one barrier where we don't see many samples to be reused. Because again, if you think about the cost is high. It's around 50, for example, it's 50 cents per sample to run alignment on a typical RNA-seq. But multiply that by 80,000 samples being there, you can do the math and you can see the price is high to reuse all the public data. Okay. Yeah, thank you.
Very nice.
Yep, thank you.
Thank you.